Welcome to part 2 of this webinar series on systematic reviews and meta-analyses. In part 1, I reviewed what SRMAs are, their purpose, and the importance of combining a systematic review with a meta-analysis. In this session, I will review the steps in conducting the systematic review, and then in part 3, we will cover the conduct of the meta-analysis. My only disclosure is that I am currently a MyTax Elevate postdoctoral fellow jointly funded by the Government of Canada and the Canadian Sugar Institute. With the conduct of the systematic review, I will go over 8 general steps. It is important to understand the steps in how a systematic review is conducted for you to be able to critically assess a SRMA you are reading. So the first step is the formulation of the research question. The reason why the research question is so important is because if you don't develop an appropriate research question and search criteria, the studies you include can give you an overall conclusion which may not be true or meaningful. So the answer you get from your meta-analysis will depend on the question you ask. The question will define the types of studies you will include, the outcomes you will look at, and will define the exposure or intervention. Most importantly, it will define the generalizability of the conclusions which you draw from your analyses. Formulating your research question can be guided by using the PCOTS framework. This will help you to define the eligibility criteria for the studies which will be included. In defining the research question, consider that specific questions can be explored in subgroup and dose response analyses. Using the PCOTS acronym will help in developing a good clinical question to ensure that essential components are identified. These include the population of interest, the intervention or exposure you want to explore, the comparator that you want to be comparing your intervention to, what the outcome variable is that is affected by your intervention, the time for follow-up or duration of the intervention, and the study design, for example, limiting your search to randomized controlled trials. Here is an example of a template that can be used to construct a research question for an intervention. Now here is an example for a debate regarding pasta consumption and obesity, where current public messages seem to tell us that eating pasta can make you gain weight. We recently addressed this debate in the design of a SRMA using the PCOTS framework, in which we identified that the population we were interested in was adults as opposed to children. The intervention was pasta consumption. The comparator was any other carbohydrate food, for example, bread or potatoes. The outcome was body weight. The time was a follow-up greater than 3 weeks, and the study design was randomized controlled trials. So our research question was, in adults, how does pasta consumption compare to other carbohydrates in the effect on body weight within randomized controlled trials greater than 3 weeks? This gives you a good basis to begin thinking about the types of studies you want to include in your systematic review and meta-analysis. Take a second and think about what the exclusion criteria would be for this SRMA. Some examples of exclusion criteria include children, acute studies less than 3 weeks, and trials that are not randomized. Other aspects that should be considered both in designing a SRMA or reading one include which variations in interventions can be combined. For example, if you are interested in oat fiber specifically and would want to restrict studies you include to those that provide some amount of oat beta-glucan, would you also want to consider whether or not other oat products should be included? For example, those that are maybe more highly processed potentially do not include beta-glucan. Consider if there are any health claims for your nutrient or food of interest, and whether you would want to be more specific in the studies you include to be addressing a question that falls in line with a guideline already in place. If, for example, studies with the more highly processed oat product, perhaps without beta-glucan fibers, are included, they may not be as effective and thus will impact the conclusions you draw in your analysis. Another consideration is which tools for outcome measurement can be combined. Sometimes there are various ways in which an outcome can be measured, which can impact the variance or reliability of that measure. For example, if your outcome is body weight, would you want to include studies in which body weight was self-reported by participants, or would you want to only include those studies in which body weight was measured by study staff in clinic following a standard protocol where shoes are removed and it is done using the same scale throughout the study for all participants? Other examples include measurements which can be done using different methods, like either in blood samples or urine samples. Consider whether or not it is justified to combine these studies, or is the method in one bettered? For example, is it a gold standard? A further consideration is which populations can be combined. Consider whether your intervention can have different effects in different population types. For example, those that are generally healthy versus those with some diagnosed chronic condition. Would it be appropriate to combine these studies? If there is no known evidence that the effect may differ among different patient types, you may want to include studies of all health backgrounds and then perform subgroup analyses to explore whether there are any interactions between different patient types. Once you have a good sense of your research question, you will want to perform a quick search of the literature using broad keywords in, for example, PubMed, with some study restrictions. If you are reviewing a SRMA on a topic, you will want to perform this step to compare it to other SRMAs which have been done and assess how it differs to ensure you have not only the most up-to-date one, but also the one that is most relevant to the question you are looking for. After all these considerations, you will have a good idea of the eligibility criteria for the studies which will be included as part of the systematic review. Here is an example of the criteria used from the PASTA and body weight SRMA in randomized controlled trials I discussed earlier. Once the research question is developed and eligibility criteria set, the next step is to think of the a priori hypotheses which may explain potential heterogeneity in the studies which will be included. Heterogeneity may be an unknown word for you, but think of it as the variability or differences between studies. I will explain more on heterogeneity later. The a priori hypotheses will be used to conduct subgroup analyses in the meta-analysis. By a priori, we mean that they are thought of during the stage of the development of the protocol, or prior to study commencement. Think back to the Dr. Black and Dr. White example from part 1. The set of 10 studies that Dr. Black included were very different from the set of 10 studies included by Dr. White. Why? Our a priori identification of reasons why our studies may differ will help to explain the differences we may see in our meta-analysis. Because subgroups are used to try to explain variability in the results between studies, it is important to think of aspects within which your studies may vary and which may cause some biologically plausible difference in the effect. This is where the expertise of the person conducting the SRMA comes into play. Here are some examples of a priori hypotheses that could be used. They include design, for example, looking at studies that are crossover in design compared to parallel studies. Looking at the example of dose, you would have to decide what cut points you would want to assess in your subgroups. In nutrition trials, the dose or amount of that nutrient explored in various trials may vary quite widely. Therefore, thinking about a dose subgroup would be a very important consideration. The cut point could be the median intake in the population if this is known, or it could be a cut point outlined in a guidance for a minimum or maximum suggested intake, since within the guideline, it will already explain the biological plausibility for the difference in dose. This is important to consider because in studies where the intervention was consumed at a very low dose, there may be no effect, but as the dose increases, an effect may be seen. Studies can also arrive at different results because of the quality of the study and the type of population that was recruited. These a priori hypotheses of ways in which studies may differ because of some plausible difference is important to consider and to look for in SRMAs, as they may significantly impact how you understand the results and draw your conclusions. Once the above parameters are in place, a protocol for the SRMA is pretty much in place. It is important that protocols for SRMAs are published on a publicly available database so that you have transparency in what the original plan was and that the conduct followed a well thought out and predetermined plan to minimize bias, just as with any other original piece of research. One last piece of the protocol I haven't yet discussed is the methodology. Guidelines on how to properly conduct SRMAs can be found in the Cochrane Handbook for Systematic Reviews of Interventions. This handbook can be found online. There is also a nicely laid out document to follow for reporting of SRMAs to ensure that all important aspects are not only conducted, but reported for completeness. Many journals have the completion of the PRISMA checklist as a requirement for publication. PRISMA is an evidence-based minimum set of items for reporting in systematic reviews and meta-analyses. We will review this document in a subsequent video, but each item is actually covered in today's webinar. Once the protocol is complete, the search can be conducted. Systematic reviews are at risk of presenting misleading results if they fail to secure a complete or representative sample of the available eligible studies. There are 3 important points to consider when we are conducting our systematic search. To secure a complete or representative sample of the available eligible studies, for most clinical questions, searching a single database is insufficient. Thus, the first is to select multiple databases to conduct the search in order to ensure that we capture all the studies out there on the topic. The 3 databases listed here as examples include Medline, Embase, and the Cochrane Library. All database searches should be supplemented with manual searches of the reference lists of all selected articles, as well as review articles. This allows for a complete search and retrieval of all eligible studies. The next important step is to have a well-developed set of search terms. Again, this is important in order to ensure to capture all the studies that are eligible on the topic in order to properly answer the research question. In developing the search terms, it is encouraged to schedule an appointment with a librarian to ensure that the search terms allow for a broad capture of studies on the topic without being excessively large. Let's think of the pasta example. How can we ensure to capture all studies looking at the effects of pasta? If I just search the word pasta, will I get all studies? Are there other terms that I should use instead of just the word pasta? How about spaghetti, macaroni, or lasagna? It is important to consider the different ways your intervention may be expressed in the literature in order to ensure that you capture them in your search terms. Here I have an example of the search terms used in a systematic review of glycemic index and body weight. Additional considerations include thinking of the different ways that your search words can be spelled, the different potential endings of the words, and the variety of ways to capture the outcome. For example, body weight may be reported in studies that do not have body weight explicitly written in the title or abstract. For example, studies on body mass index may have body weight results in the report. When conducting the search, the use of any language restrictions should be avoided. For any non-English studies, one can consult a library to see if they have translational services. These steps will help to ensure a comprehensive search strategy to minimize missing relevant studies and thus minimize the potential for misleading results. After completing the search, the previously established eligibility criteria are applied to all articles retrieved from the search. 2 independent reviewers will screen through the titles and abstracts, and for those studies which pass this stage, they will then review the full text and again apply the eligibility criteria. The final set of studies deemed eligible for inclusion will then have all data extracted. This is an example of a data extraction form used for each eligible study. This is a printable example, but all information will be entered into online software. Aside from extracting data on the endpoints of interest, data on each of the a priori subgroups determined earlier is collected, including study design, population type, details on the intervention, as well as the control, including dose. This is done by 2 independent reviewers who then compare their forms, and if there are any discrepancies, a 3rd person will be consulted. These are the most tedious steps of conducting a SRMA. Another important assessment is that of study quality, which pertains to the quality of each individual study included in the SRMA. There are a few different tools which can be used to assess study quality depending on the type of study. However, all tools are assessing areas at which bias may influence a study and thus its quality. These include different aspects of trial design, conduct, and reporting. When it comes to controlled trials, examples of the areas which are assessed for bias include the following, like randomization and whether there is missing outcome data. For example, if the sample size analyzed differs from that randomized, which then introduces an influence of bias since the benefits of randomization may be lost. Through the study quality assessment, each individual study in the SRMA is assessed based on each of the risk of bias domains using what is reported in the study. This is an example of the Cochrane risk of bias tool, which is used for randomized controlled trials. You can see that there are a set of criteria used to assess each of the categories of risk. Each category is assessed as having either low, high, or unclear risk of bias. However, these tools are periodically updated. For example, this tool itself was just updated, and there may be slight differences in rating, which you may notice for newer SRMAs. So here's the update on the Cochrane risk of bias tool, now called ROB2. This is what it looks like. It includes an electronic step-by-step process for evaluating each risk of bias domain. When it comes to observational studies, like cohort studies or case control studies, which can be included in a SRMA, the Newcastle Ottawa score is an example of a tool which can be used to assess study quality, as shown here. So that sums up how risk of bias can be and should be assessed for each individual study in a SRMA in order to obtain an assessment of the quality of the included studies. Once all the data is extracted and there is agreement between the independent reviewers, the meta-analysis can be performed. I just want to highlight one last important piece to understand. Once you are ready to perform the meta-analysis, the statistics will be run using the data provided. So if the systematic review was not well thought out and identified a set of studies which do not capture all the data out there on a topic, or are too broad, and that the data are like mixing apples and oranges, then the meta-analysis will generate a result which is not generalizable or applicable. Further, if the ROB or risk of bias assessments are performed, as they should, and reveal that the majority of studies are of poor quality, this too indicates that we may not have confidence in the results of the meta-analysis. This is what has been termed garbage in equals garbage out. Therefore, it is a great skill to understand these general steps in the conduct of a systematic review, so you know what to look for when reading ASRMA and make you more confident in your ability to critically assess it and interpret the results for yourself. We will review these general steps again in a subsequent webinar when we use them to help us critically assess ASRMA. But up next is how the meta-analysis is performed. So look forward to part 3. I'd like to acknowledge my mentors and lab group, as many of these slides have been developed and shared by members of our team.